Welcome to Sleepy Science Hub. I'm so glad you found your way here tonight, to this peaceful corner where we explore Earth's most gentle mysteries together. As you settle into your most comfortable position, wherever you might be in the world right now, take a moment to let me know in the comments where you are and what time it is as you listen. There's something wonderfully grounding about knowing we're sharing this quiet moment of discovery together, connected across our planet, while we learn about the slow, patient processes that have been shaping our world for thousands of years. Tonight, we're going to journey to one of Earth's most serene and ancient landscapes, the Arctic, to understand a phenomenon that sounds dramatic but unfolds with the gentle patience of nature itself. These are methane time bombs, but don't let the name concern you. We're not exploring anything explosive or frightening. Instead, we'll discover how tiny microorganisms, working quietly in frozen soils for millennia, are part of Earth's natural breathing rhythm, slowly releasing ancient carbon back into the atmosphere in processes that connect us to climates from long ago. Now, get comfortable. Adjust your pillows, pull your blankets close, and let your breathing settle into its natural, peaceful rhythm. Take a slow, deep breath of the night air around you. Feel your body releasing the tensions of the day, and let's begin this tranquil journey to the far north, where ice and time have preserved stories written in the earth itself. As you lie there in comfort, your breathing gradually slowing and deepening like the gentle pulse of Arctic tides beneath layers of ancient ice. Imagine yourself standing on the edge of the vast northern wilderness on a calm summer evening. The sun hangs low in the sky, painting the landscape in soft shades of gold and amber that seem to stretch endlessly across rolling tundra dotted with thousands of small lakes that mirror the peaceful heavens above. This is the Arctic in its most serene state, a land where time moves differently, where seasons unfold with the patience of geological processes, and where the very ground beneath your feet holds secrets that have been accumulating for thousands upon thousands of years. The air here is crisp and clean, carrying the faint scent of Arctic willows and the fresh, mineral smell of ancient ice slowly melting in the summer warmth. As you breathe in this pure northern air, you're participating in an atmospheric dance that connects you to every living thing on the planet. The slow exchange of gases that has been cycling through Earth's systems since life first began to breathe. But here in the Arctic, this breathing takes on a particularly profound character, as the landscape itself seems to inhale and exhale with the changing seasons, storing carbon for millennia during the long, cold winters and gently releasing it during the brief, luminous summers. Beneath your feet, hidden under a thin layer of summer-active soil, lies one of Earth's most remarkable natural archives, the permafrost. This permanently frozen ground extends downward for hundreds of meters in some places, creating a vast underground library where plant and animal remains from ancient climates have been preserved in a state of suspended animation for tens of thousands of years. The permafrost holds within its icy embrace the carbon from entire forests that grew when the Earth was warmer, from grasslands that stretched across what is now frozen ground, and from countless generations of plants and animals that lived and died when the Arctic was a very different place. As your consciousness settles into harmony with this ancient landscape, your breathing becoming as steady and measured as the seasonal rhythms that govern Arctic life, you begin to understand that the permafrost represents one of nature's most patient processes. For millennia, this frozen ground has been slowly accumulating organic matter, layer by layer, season by season, creating a vertical timeline that climate scientists can read like the pages of Earth's atmospheric history. Each layer tells a story of what the climate was like when it was deposited, preserving pollen, seeds, leaves, and even entire animals in a state of perfect preservation that allows scientists to peer back into Earth's climatic past. The formation of permafrost happened gradually, over thousands of years, as global temperatures cooled and the ground froze deeper and deeper each winter, eventually reaching depths where the summer warmth could no longer penetrate. This process created a kind of natural deep freeze that has been operating continuously in some areas for over 700,000 years, making Arctic permafrost older than our species and preserving organic matter from climates that existed long before humans walked the Earth. The permafrost became a vast carbon sink, locking away atmospheric carbon dioxide that had been absorbed by ancient plants and storing it in the frozen ground like a massive underground vault. The landscape you're experiencing is shaped by this hidden underground ice in ways both subtle and profound. The small lakes the tundra, called thermokarst lakes, form when ground ice melts and creates depressions that fill with water. These lakes create their own microclimates, staying slightly warmer than the surrounding land and often remaining unfrozen even during the harsh Arctic winter. Around their edges, you can sometimes see small bubbles rising through the water, a gentle sign of the chemical processes happening in the sediments below, where ancient organic matter is slowly being transformed by microorganisms that have learned to live and work in environments where oxygen is scarce. The vegetation that grows here has adapted to life above the permafrost in remarkable ways. Arctic willows grow close to the ground, their roots spreading horizontally rather than diving deep, since the permanently frozen layer prevents deep root growth. Mosses and lichens form soft carpets that help insulate the ground, keeping the permafrost frozen even during the warm summer months. These plants are part of a delicate balance that has existed for thousands of years, where the living vegetation helps protect the frozen organic matter beneath from thawing too rapidly. The wildlife that calls this landscape home, caribou, Arctic foxes, countless species of birds, are all connected to the permafrost ecosystem in ways that demonstrate the intricate relationships between life and landscape in the Arctic. Caribou migrate across vast distances, following ancient routes that take advantage of the firm ground provided by permafrost, while their grazing patterns help maintain the vegetation communities that help keep the permafrost stable. The timing of their migrations, the plants they eat, and even the nitrogen they deposit through their waste all become part of the complex web of interactions that influence how carbon moves through the Arctic system. The indigenous peoples who have lived in the Arctic for thousands of years have developed deep knowledge of how to read the subtle signs of permafrost behavior. They know which areas provide the most stable ground for building, which seasons are safest for travel across frozen lakes, and how the changing patterns of ice formation reflect broader changes in the Arctic climate system. Their traditional knowledge represents generations of careful observation of how the landscape responds to natural variations in temperature and precipitation, creating a living library of Arctic climate understanding that complements modern scientific investigation. The scientific research stations scattered across the Arctic represent humanity's more recent attempt to understand the complex processes occurring in permafrost regions. These installations, often small clusters of buildings connected by wooden walkways that minimize disturbance to the underlying permafrost, house instruments that continuously monitor ground temperatures, atmospheric gas concentrations, and the subtle movements of the soil as it freezes and thaws with the seasons. The data they collect reveals that the Arctic is experiencing changes more rapid than almost anywhere else on Earth, with temperatures rising at twice the global average and creating conditions that are beginning to affect the stability of permafrost that has remained frozen for millennia. The summer measurements taken by these research stations show that the active layer, the surface layer of soil that thaws each summer and refreezes each winter, has been growing deeper in recent decades. Where once only the top few centimeters of soil might thaw during a brief Arctic summer, now the thaw can penetrate a meter or more into previously frozen ground. This deeper thawing exposes organic matter that has been frozen for thousands of years to warmer temperatures and to the microorganisms that can begin the process of decomposition. The permafrost research reveals that this frozen ground contains an enormous amount of carbon, more carbon than currently exists in the entire atmosphere. This carbon is stored in the form of partially decomposed plant and animal matter that accumulated during past warm periods and was then preserved when the climate cooled and the ground froze. The preservation is so complete that scientists regularly find intact leaves, seeds, and even the remains of large mammals like woolly mammoths in permafrost cores, testament to the remarkable preservative power of the frozen ground. The chemical composition of permafrost shows that it contains not just carbon, but nitrogen, phosphorus, and other nutrients that were part of ancient ecosystems. When permafrost thaws, these nutrients become available to modern plants and microorganisms, creating a complex feedback system where ancient ecosystems influence contemporary ones. The release of these nutrients can stimulate plant growth in some areas, leading to increased carbon absorption from the atmosphere, while in other areas, the nutrients fuel microbial activity that releases carbon back to the atmosphere. The seasonal cycles that govern permafrost behavior create a natural rhythm of expansion and contraction, freezing and thawing, that shapes every aspect of Arctic ecosystems. During the long winter, when temperatures can drop to -40 degrees Celsius or lower, the ground freezes solid to great depths, and biological activity slows to nearly nothing. The landscape becomes locked in crystalline stillness, with even the movement of water stopping as lakes and streams freeze solid. But this apparent dormancy conceals ongoing chemical processes that continue even at extremely low temperatures, as microorganisms adapted to Arctic conditions continue their patient work of transforming organic matter even in the depths of winter. The spring thaw brings a dramatic transformation to the Arctic landscape, but it happens gradually over the course of several months. First, the surface snow begins to melt, creating temporary streams and pools that reflect the returning sunlight. Then the top layer of soil begins to thaw, releasing moisture that has been locked in ice crystals through the winter. This moisture supports a burst of plant growth and microbial activity that makes the brief Arctic summer remarkably productive despite its short duration. The thawing soil becomes soft and spongy, full of water that cannot penetrate the still frozen layers below. The acoustic environment of the thawing Arctic tells its own story of the processes occurring in the permafrost. You might hear the gentle dripping of meltwater, the soft crackling of ice as it releases air bubbles that have been trapped for months, and the subtle sounds of the soil settling as ice crystals melt and refreeze. In the lakes, there are often soft popping sounds as gas bubbles that have accumulated under the winter ice are suddenly released when the ice breaks up, creating a natural percussion that announces the changing season. The optical phenomena that occur in the Arctic landscape create visual cues that help scientists and indigenous observers understand what's happening below the surface. The color of lake water can indicate the types of chemical processes occurring in the sediments, while patterns of vegetation growth can reveal where permafrost is stable and where it might be beginning to thaw. Even the way snow accumulates and melts can provide information about the thermal properties of the underlying ground. As you continue to rest peacefully in this Arctic landscape, your breathing now completely synchronized with the slow, seasonal rhythms of the northern wilderness, you can begin to appreciate how the permafrost represents one of Earth's most remarkable examples of natural preservation and gradual transformation. This frozen ground has been patiently storing carbon and preserving the remains of ancient ecosystems for millennia, creating a natural archive that connects us to Earth's climatic past while quietly influencing its atmospheric future. The permafrost ecosystem demonstrates how life and landscape can work together to create stability over vast timescales, with plants, animals, microorganisms, ice, and soil all participating in a complex dance that has been ongoing for thousands of years. Yet this stability is not static but dynamic, constantly adjusting to natural variations in temperature and precipitation while maintaining its essential character through slow, patient processes that operate according to the deep rhythms of geological time. As you settle deeper into peaceful contemplation, your breathing now flowing like the gentle circulation of nutrients through frozen soil layers. Let your awareness descend beneath the surface of this Arctic landscape to discover the remarkable world that exists in the permanently frozen ground. Here, in the crystalline depths of the permafrost, lies one of Earth's most extraordinary preservation systems, a natural deep freeze that has been quietly collecting and storing organic matter for tens of thousands of years, creating vast underground reserves of carbon that tell the story of ancient climates and vanished ecosystems. Picture yourself descending slowly through the layers of permafrost, like moving through a vertical museum where each level represents a different chapter in Earth's climatic history. The journey downward through the frozen ground reveals a progression of increasingly ancient organic matter, from plant roots and leaf litter that were buried just decades ago near the surface, to the preserved remains of entire forests and grasslands that grew during warm periods thousands of years in the past. This organic matter exists in a state of suspended animation, protected by the frozen conditions from the decomposition that would normally return its carbon to the atmosphere within years or decades of the organism's death. The chemistry of this preservation process involves the transformation of ice from a simple frozen water into a complex matrix that can hold organic matter in perfect stasis for geological time periods. When plant and animal tissues become incorporated into permafrost, they don't simply freeze like food in a household freezer. Instead, they become part of an intricate ice-soil matrix where water molecules form crystalline structures that effectively mummify organic matter, preserving not just the carbon content, but often the original cellular structure, DNA, and even the chemical signatures of the atmospheric conditions that existed when the organisms were alive. As your consciousness settles into harmony with these ancient preservation processes, your breathing becoming as steady and patient as the accumulation of organic matter over millennia. You begin to understand how the permafrost has served as Earth's natural carbon vault, steadily withdrawing carbon dioxide from the atmosphere and storing it in frozen form beneath the ground. This process has been so effective that permafrost regions contain more carbon than all of Earth's forests combined, representing approximately 1,400 billion tons of carbon locked away in frozen soils across the Arctic and subarctic regions of the planet. The organic matter preserved in permafrost comes from a remarkable variety of sources that reflect the diversity of life that has existed in Arctic regions during different climatic periods. Pollen grains preserve information about the types of plants that grew in ancient warming periods, while preserved seeds can still germinate after being frozen for thousands of years, demonstrating the remarkable effectiveness of permafrost preservation. The remains of large mammals like woolly mammoths, woolly rhinoceros, and cave bears provide dramatic evidence of the rich ecosystems that once flourished in regions that are now permanently frozen, while microscopic organisms preserve information about ancient soil conditions and atmospheric chemistry. The stratification of organic matter in permafrost creates natural climate archives that scientists can read like tree rings or ice cores to understand how Arctic ecosystems have responded to past climate changes. The deeper layers, which formed during warmer periods tens of thousands of years ago, often contain the remains of boreal forests with large trees and diverse understory vegetation. Above these lie layers formed during cooler periods, containing more tundra-like vegetation and the remains of cold-adapted species. The uppermost layers reflect more recent ecosystems that developed during the current interglacial period, creating a vertical timeline that documents the dramatic ecological changes that have occurred in Arctic regions over time. The preservation quality in permafrost is so remarkable that scientists have been able to extract viable bacteria and viruses from frozen soils that are thousands of years old, demonstrating that permafrost doesn't just preserve dead organic matter, but can also maintain living microorganisms in a state of suspended animation. These ancient microbes, when thawed under laboratory conditions, can resume their biological activities as if they had been frozen just yesterday, providing unique insights into the evolution of microbial communities and the types of organisms that lived in past Arctic ecosystems. The formation process that created these vast permafrost carbon stores occurred during the late Pleistocene epoch, roughly between 60,000 and 11,000 years ago, when much of the northern hemisphere was covered by massive ice sheets. During this period, a unique ecosystem called the Mammoth Steppe covered much of Siberia, Alaska, and northern Canada. This ecosystem supported enormous herds of large mammals and was characterized by highly productive grasslands that could accumulate large amounts of organic matter each growing season. When the climate cooled and ice sheets expanded, Much of this organic matter became incorporated into developing permafrost, creating the massive carbon stores that exist today. The physical structure of permafrost varies dramatically depending on the conditions under which it formed and the types of organic matter it contains. Some permafrost is more than 80% ice by volume, creating a crystalline matrix that holds organic matter in tiny pockets between ice crystals. Other permafrost contains less ice but higher concentrations of organic matter, creating dense, black layers that can contain 40% or more carbon by weight. The distribution of this organic matter is not uniform, but forms distinct layers and pockets that reflect the ecosystem conditions that existed when each layer was deposited. The age dating of permafrost organic matter reveals that some carbon has been frozen for extraordinary lengths of time. Radiocarbon dating shows that organic matter in the deepest permafrost layers can be 40,000 to 50,000 years old, representing carbon that was removed from the atmosphere during the middle of the last ice age. Some permafrost deposits are too old for radiocarbon dating and may contain organic matter that is hundreds of thousands of years old, making them among the oldest preserved organic materials on Earth outside of fossil deposits. The chemical composition of permafrost organic matter provides detailed information about the atmospheric conditions that existed when ancient plants were growing. The isotopic ratios of carbon in preserved plant material reflect the concentration of carbon dioxide in the ancient atmosphere, while nitrogen isotopes provide information about soil conditions and the availability of nutrients. These chemical signatures allow scientists to reconstruct not just what types of plants were growing in ancient Arctic ecosystems, but also what the climate and atmospheric chemistry were like at different times in the past. The microbial communities that exist within permafrost represent some of Earth's most remarkable examples of life in extreme environments. These microorganisms have adapted to survive in conditions of permanent freezing, extremely low nutrient availability, and complete absence of sunlight. Some permafrost microbes can remain active at temperatures as low as minus 20 degrees Celsius, carrying out slow but measurable metabolic processes even in frozen conditions. These cold-adapted organisms play crucial roles in the chemical cycling that occurs within permafrost, slowly transforming organic matter and influencing the types of gases that are produced when permafrost eventually thaws. The vertical distribution of different types of organic matter in permafrost reflects the changing environmental conditions that occurred during its formation. The upper layers typically contain more recently deposited organic matter from tundra vegetation, with high concentrations of moss, sedge, and low shrub remains. Deeper layers often contain more woody material from trees and large shrubs that grew during warmer periods, while the deepest layers may contain organic matter from grassland ecosystems that supported large herbivore populations. This vertical zonation creates distinct biogeochemical environments within the permafrost that influence how the organic matter will behave when thawing occurs. The moisture content of permafrost organic matter plays a crucial role in determining how it will decompose when thawed. Organic matter that was deposited in dry conditions tends to be better preserved and may decompose more slowly when thawed, while organic matter that was deposited in wet conditions often contains more labile carbon compounds that can be rapidly processed by microorganisms when thawing occurs. The moisture history recorded in permafrost deposits provides information about past precipitation patterns and hydrological conditions in Arctic regions. The preservation of atmospheric gases within permafrost creates natural archives of ancient air composition. Tiny bubbles trapped in permafrost ice can contain samples of ancient atmosphere, while organic matter preserves chemical signatures that reflect the atmospheric conditions under which plants grew. These atmospheric archives provide independent confirmation of ice core records and help scientists understand how atmospheric chemistry has changed over time in response to natural climate variations. The interaction between permafrost organic matter and mineral soil components creates complex chemical environments that influence both preservation and decomposition processes. Iron and other metals in permafrost soils can form chemical complexes with organic compounds that enhance preservation, while the pH and nutrient content of permafrost influence the types of microbial communities that can survive in frozen conditions. These soil organic matter interactions create feedback systems that can either stabilize organic carbon in permafrost or make it more susceptible to decomposition when thawing occurs. The regional variation in permafrost organic matter content reflects the diverse environmental conditions that exist across Arctic and subarctic regions. Permafrost in Siberia tends to contain extremely high concentrations of organic matter due to the productive mammoth steppe ecosystems that existed there during the Pleistocene, while permafrost in other regions may contain lower but still significant amounts of organic carbon. These regional differences influence how different permafrost regions will respond to climate warming and what types of greenhouse gases will be produced as thawing progresses. The layered structure of permafrost organic matter creates distinct microenvironments that support different types of microbial communities and chemical processes. Aerobic conditions near the surface support microbes that require oxygen, while deeper anaerobic layers support entirely different communities of microorganisms that have evolved to survive without oxygen. These different microbial communities produce different end products when they decompose organic matter, influencing whether carbon is released as carbon dioxide or methane when permafrost thaws. The study techniques that scientists use to analyze permafrost organic matter must account for the fact that thawing the samples can immediately begin decomposition processes that alter the original characteristics of the preserved material. Researchers have developed specialized methods for studying permafrost that keep samples frozen during analysis or that can measure chemical processes at sub-freezing temperatures. These techniques have revealed that permafrost is not simply an inactive carbon store, but a dynamic system where slow chemical and biological processes continue even under frozen conditions. The connection between permafrost organic matter and global carbon cycling demonstrates how local preservation processes can have planetary scale implications. The carbon stored in permafrost represents a significant fraction of all the carbon in terrestrial ecosystems, and changes in permafrost stability can influence atmospheric greenhouse gas concentrations and global climate patterns. This connection between local Arctic processes and global atmospheric chemistry illustrates how Earth's climate system integrates processes occurring across vastly different spatial and temporal scales. As you continue to rest peacefully within this frozen archive of Earth's climatic past, your breathing now perfectly synchronized with the patient accumulation processes that have been storing carbon in permafrost for thousands of years, you can appreciate how these ancient sleepers beneath the Arctic landscape represent one of nature's most remarkable examples of long-term carbon management. The organic matter preserved in permafrost connects us directly to ancient ecosystems and climate conditions, while quietly influencing the atmospheric chemistry that will shape Earth's future climate through the slow, patient release of ancient carbon back into the modern atmosphere. As you sink deeper into tranquil understanding, your breathing now flowing like the gentle metabolic rhythms of microscopic life working in the depths of thawing permafrost. Let your awareness turn to some of Earth's most patient and persistent inhabitants, the methane-producing microorganisms that serve as tiny architects of atmospheric change. These remarkable creatures, invisible to the naked eye yet capable of transforming the chemistry of entire landscapes, represent one of life's most ancient and successful strategies for extracting energy from environments where oxygen is scarce and traditional forms of respiration cannot occur. Picture yourself shrinking down to enter the microscopic world that exists in the waterlogged sediments and thawing permafrost of Arctic landscapes, where billions of microorganisms work together in communities so vast and productive that their collective activities can influence the composition of Earth's atmosphere. Here, in the oxygen-free depths of saturated soils and lake sediments, these tiny engineers have been quietly carrying out one of the most important biogeochemical processes on our planet for billions of years, transforming organic matter into methane through a series of elegant biochemical reactions that demonstrate life's remarkable ability to find energy sources in even the most challenging environments. The methane-producing microorganisms, known scientifically as methanogens, represent some of the most ancient forms of life on Earth, with evolutionary lineages that trace back nearly 4 billion years to a time when Earth's atmosphere contained very little oxygen, and early life forms had to find alternative ways to extract energy from their environment. These microbes belong to a domain of life called archaea, which are distinct from both bacteria and the more complex cells that make up plants and animals. Their ancient origins and unique biochemistry allow them to thrive in environments that would be hostile to most other forms of life. As your consciousness settles into harmony with these microscopic processes, your breathing becoming as steady and rhythmic as the metabolic cycles that govern microbial life, you begin to understand how methanogens have evolved sophisticated biochemical pathways that allow them to break down organic matter in the complete absence of oxygen. This process, called methanogenesis, involves a complex series of chemical reactions where organic compounds are systematically dismantled and reconstructed, with methane gas as the final end product that gets released into the surrounding environment. The environments where methanogens thrive are characterized by their lack of oxygen, abundant organic matter, and often high moisture content, conditions that are perfectly represented in the thawing permafrost and waterlogged soils of Arctic regions. These anaerobic environments develop when organic matter accumulates faster than it can be decomposed by oxygen-requiring organisms, creating zones where all available oxygen is quickly consumed and only organisms capable of anaerobic metabolism can survive. The transition from aerobic to anaerobic conditions happens gradually, often over just a few centimeters of depth in saturated soils, creating distinct biogeochemical zones with very different microbial communities and chemical processes. The community structure of methanogenic ecosystems reveals a remarkable example of biological cooperation, where different groups of microorganisms work together in sequential steps to completely break down complex organic matter. The process begins with hydrolytic bacteria that break down large organic molecules like cellulose and proteins into smaller compounds. These smaller molecules are then processed by fermentative bacteria that convert them into simple organic acids, alcohols, and hydrogen gas. Finally, the methanogens use these simple compounds as energy sources, converting them into methane and carbon dioxide through their unique metabolic pathways. The biochemistry of methanogenesis involves some of the most unusual chemical reactions found in living systems, utilizing enzymes and cofactors that are unique to archaeal organisms. The process requires strict anaerobic conditions because the enzymes involved in methane production are extremely sensitive to oxygen and become inactive if exposed to even trace amounts of this gas. This sensitivity to oxygen explains why methanogenesis only occurs in environments where oxygen has been completely depleted, such as the waterlogged sediments of Arctic lakes or the saturated zones of thawing permafrost. The different pathways of methanogenesis allow these microorganisms to utilize various types of organic substrates, depending on what's available in their environment. Some methanogens specialize in converting simple compounds like acetate directly into methane and carbon dioxide, while others can use hydrogen gas and carbon dioxide to produce methane through a process that essentially reverses combustion. Still others can break down more complex organic compounds like alcohols and organic acids, demonstrating the metabolic flexibility that has allowed these organisms to colonize such a wide range of anaerobic environments. The growth rates of methanogenic microorganisms are typically much slower than those of organisms that use oxygen for respiration, reflecting the lower energy yield of anaerobic metabolism. While aerobic bacteria might double their population in hours, methanogens often require days or weeks to complete a single generation cycle. This slower metabolism means that methanogenic processes develop gradually, often taking months or years to reach full activity in newly thawed permafrost. However, once established, these communities can remain active for decades, steadily producing methane as long as suitable conditions persist. The temperature sensitivity of methanogens plays a crucial role in determining how much methane is produced as Arctic regions warm. These microorganisms can remain active at surprisingly low temperatures, with some continuing to produce methane even in nearly frozen conditions. However, Their activity increases dramatically as temperatures rise, following exponential curves that mean small increases in temperature can lead to large increases in methane production. This temperature sensitivity creates a powerful feedback mechanism where warming temperatures stimulate increased methane production, which can contribute to further atmospheric warming. The substrate preferences of different methanogenic species influence the types of organic matter that get converted to methane when permafrost thaws. Some methanogens specialize in processing fresh plant material, converting recently deposited leaf litter and root material into methane relatively quickly. Others are adapted to work with older, more decomposed organic matter, slowly breaking down carbon compounds that have been partially processed by other microorganisms. The diversity of methanogenic species in thawing permafrost means that both fresh and ancient organic matter can serve as sources for methane production. The water chemistry of methanogenic environments plays a critical role in determining which species can thrive and how much methane gets produced. The pH, salinity, and nutrient content of soil water all influence methanogenic activity, with different species adapted to different chemical conditions. Some methanogens prefer neutral pH conditions, while others can tolerate acidic or alkaline environments. The availability of nutrients like nitrogen and phosphorus can also limit methanogenic activity, creating complex interactions between soil chemistry and microbial metabolism. The spatial distribution of methanogenic activity in thawing permafrost creates distinct zones with different levels of methane production. The highest activity typically occurs in the zone where permafrost is actively thawing, where freshly thawed organic matter encounters established populations of methanogens. Lower activity zones exist in areas where organic matter has already been partially processed, while inactive zones occur where conditions are still too cold or too dry for significant methanogenic activity. This spatial heterogeneity means that methane production is patchy across Arctic landscapes, with hotspots of high activity surrounded by areas of lower production. The seasonal patterns of methanogenic activity reflect the strong influence of temperature and moisture on these biochemical processes. During the brief Arctic summer, when soil temperatures are highest and thaw depth is greatest, methanogenic activity reaches its peak, producing the majority of annual methane emissions. During winter, when soils refreeze and biological activity slows dramatically, methane production drops to very low levels, though it doesn't stop entirely. This seasonal cycling creates pulses of methane release that coincide with the warmest periods of the year. The interaction between methanogens and other soil microorganisms creates complex competitive and cooperative relationships that influence overall methane production. Methanogenic bacteria must compete with other anaerobic bacteria for available organic substrates, while also depending on these same bacteria to break down complex organic matter into forms they can use. The balance between these different microbial groups can shift in response to environmental changes, influencing whether organic matter gets converted to methane or to other end products like organic acids or carbon dioxide. The adaptation mechanisms that allow methanogens to survive in permafrost environments include the production of antifreeze proteins, specialized cell membrane compositions that remain flexible at low temperatures, and the ability to enter dormant states during periods when conditions are unsuitable for active metabolism. Some methanogens can form resistant spores that can survive freezing and thawing cycles, while others can adjust their metabolism to continue functioning at extremely low activity levels during cold periods. The laboratory studies of methanogenic activity in permafrost samples reveal that these microorganisms can remain viable in frozen conditions for thousands of years, resuming activity immediately when samples are thawed under appropriate conditions. This remarkable survival ability means that ancient methanogenic communities preserved in deep permafrost can become active contributors to methane production when climate warming causes deep thaw to occur. The reactivation of these ancient microbial communities represents a direct connection between past and present atmospheric chemistry. The molecular techniques used to study methanogenic communities in permafrost have revealed enormous diversity in the types of organisms present and their metabolic capabilities. DNA sequencing of permafrost samples shows that methanogenic communities often contain dozens of different species, each adapted to slightly different environmental conditions or specialized for processing different types of organic substrates. This diversity provides resilience to environmental changes and ensures that methanogenic activity can continue across a wide range of conditions. The stable isotope signatures produced by different methanogenic pathways provide valuable tools for scientists to trace the sources and mechanisms of methane production in Arctic environments. Different metabolic pathways produce methane with distinct carbon isotope ratios, allowing researchers to determine whether methane emissions are coming from fresh plant material, ancient permafrost carbon, or other sources. These isotopic fingerprints help scientists understand how methane production changes as permafrost thaws and different organic matter sources become available. The cultivation of methanogens from permafrost environments has revealed new species and metabolic pathways that were previously unknown to science. Many of these organisms have unique adaptations to cold temperatures and the specific chemical conditions found in permafrost, making them valuable subjects for research into extremophile biology and potential biotechnology applications. The discovery of new methanogenic pathways also helps scientists better understand and model the biogeochemical processes that control methane production in thawing permafrost. The ecological interactions between methanogens and higher organisms create complex food webs where microbial methane production supports other forms of life. Some bacteria can consume methane as an energy source, creating zones where methane is produced and consumed in close proximity. Plants growing in methanogenic soils may obtain some of their carbon from methane-derived compounds, while soil invertebrates can influence methanogenic activity through their effects on soil structure and organic matter distribution. The role of methanogens in global biogeochemical cycles extends far beyond their immediate effects on methane production, as these organisms also influence the cycling of carbon, nitrogen, sulfur, and other elements in anaerobic environments. Their activities help maintain the chemical balance of wetland ecosystems and influence the availability of nutrients for other organisms. The study of methanogens in permafrost environments provides insights into how these important biogeochemical processes may change as Arctic regions continue to warm. As you continue to rest peacefully in this microscopic world of patient biological engineering, your breathing now perfectly synchronized with the gentle metabolic rhythms that govern methanogenic activity. You can appreciate how these tiny architects of change represent one of life's most successful strategies for extracting energy from challenging environments while simultaneously influencing the atmospheric chemistry that affects our entire planet. Their ancient wisdom and remarkable adaptability demonstrate how the smallest forms of life can have the most profound effects on Earth's environmental systems. As you drift deeper into peaceful understanding, your breathing now flowing like the gentle transition of matter from solid to liquid that characterizes the Arctic's changing seasons. Let your awareness turn to one of nature's most fundamental transformations, the gradual process by which permafrost ice becomes water, setting in motion a cascade of chemical and biological changes that ripple through Arctic ecosystems like waves spreading across a calm lake. This thawing process unfolds not as a dramatic melting, but as a patient, methodical transformation that can take years or decades to complete, demonstrating how even the most significant environmental changes often happen with the quiet persistence of natural processes operating on their own timescales. Picture yourself standing beside a small Arctic lake on a warm summer morning, watching as the permafrost along its edges begins its annual thaw cycle. The process starts subtly, almost imperceptibly, as the sun's energy penetrates the dark surface soil and begins to warm the ground beneath. You might first notice it as a slight softening of the soil underfoot, or as small patches of ground that feel different when you walk across them. The ice crystals that have held the soil structure together for years or decades begin to release their grip, allowing water to move freely through spaces that have been locked solid throughout the long Arctic winter. The physics of permafrost thaw involves a complex interplay between heat transfer, moisture movement, and soil chemistry that creates conditions favorable for the biological processes that will eventually produce methane. As ground temperatures rise above freezing, ice within the soil matrix begins to melt, but this melting doesn't happen uniformly. Instead, it proceeds through the soil according to the distribution of ice, the thermal properties of different soil components, and the pathways available for heat transfer. Some areas may thaw quickly due to their exposure to solar radiation or their mineral composition, while other areas remain frozen for weeks or months longer. The thermal dynamics of thawing permafrost create distinct temperature gradients within the soil that influence how biological and chemical processes develop as thaw progresses. The surface layers, which receive direct solar heating, warm first and begin the transition from frozen to unfrozen conditions. Below this lies a transition zone, where temperatures hover around the freezing point for extended periods, creating conditions where ice and liquid water coexist in a delicate balance. Deeper still lies the permafrost table, the boundary between the seasonally thawed active layer and the permanently frozen ground below. As your consciousness settles into harmony with these gentle thermal processes, your breathing becoming as steady and gradual as the heat flow that drives permafrost thaw, you begin to understand how the transition from ice to water represents far more than just a physical phase change. When permafrost ice melts, it releases organic matter that has been physically protected from decomposition by its frozen state, suddenly making this ancient carbon available to the communities of microorganisms that specialize in breaking down organic matter under anaerobic conditions. The hydrology of thawing permafrost creates unique environmental conditions that favor the development of methanogenic processes. As ice melts, it often creates waterlogged conditions because the still frozen permafrost below acts like an impermeable barrier that prevents meltwater from draining away. These saturated conditions quickly become anaerobic, as any dissolved oxygen is rapidly consumed by aerobic microorganisms, creating the oxygen-free environment that methane-producing bacteria require for their metabolism. The result is the formation of anaerobic microsites where methanogenesis can begin to occur. The mechanical effects of freeze-thaw cycles on soil structure create additional pathways for water movement and gas transport that influence how methane production and emission develop in thawing permafrost. When water freezes, it expands, creating pressure that can fracture soil aggregates and create new pore spaces. When this ice subsequently melts, it leaves behind a changed soil structure with altered porosity and permeability. These physical changes can create preferential pathways for water movement and gas transport that influence how effectively methane can move from its production sites in anaerobic soil zones to the atmosphere. The chemical changes that accompany permafrost thaw involve complex interactions between newly mobile water, previously frozen organic matter, and the mineral components of Arctic soils. As permafrost melts, it releases not just organic carbon, but also nutrients like nitrogen and phosphorus that have been locked in frozen organic matter for thousands of years. The release of these nutrients can stimulate biological activity and influence the balance between different types of microorganisms that compete for available resources in thawing soils. The temporal patterns of permafrost thaw reflect the seasonal rhythm of Arctic climate, but with important lags and feedbacks that create complex dynamics in how thaw depth and duration change over time. The spring thaw typically begins at the surface and progresses downward as warm weather continues, but the rate and extent of thaw depend on factors like air temperature, solar radiation, precipitation, and the thermal properties of the soil. In some years, thaw may penetrate only a few centimeters into the permafrost, while in exceptionally warm years, thaw can reach depths of several meters. The spatial variability of permafrost thaw creates a mosaic of different environmental conditions across Arctic landscapes, with some areas experiencing rapid and extensive thaw while nearby areas remain relatively stable. This spatial heterogeneity reflects differences in soil type, vegetation cover, topography, and local hydrology that influence how heat transfer occurs and how effectively thaw processes can proceed. Areas with dark, heat-absorbing surfaces tend to thaw more rapidly than areas with light-colored vegetation or snow cover, while north-facing slopes may remain frozen much longer than south-facing exposures. The vegetation responses to permafrost thaw create feedback mechanisms that can either accelerate or slow continued thawing depending on the types of plant communities that develop in newly thawed areas. Some vegetation types, like dense shrubs or trees, can provide insulation that protects underlying permafrost from summer warming. Other vegetation types, like sparse grasses or bare soil, may allow more heat penetration and accelerate continued thaw. These vegetation feedbacks operate over timescales of years to decades, creating long-term trends in permafrost stability that depend on ecological as well as climatic factors. The development of thermokarst features represents one of the most visible consequences of extensive permafrost thaw, creating landscape changes that can dramatically alter local hydrology and biogeochemistry. When permafrost with high ice content thaws, the melting ice can cause the ground surface to subside, creating depressions that collect water and form new lakes or wetlands. These thermokarst features create ideal conditions for methanogenic activity because they combine abundant organic matter from thawed permafrost with the waterlogged, anaerobic conditions that methane-producing bacteria require. The geochemical processes that occur during permafrost thaw involve complex interactions between organic and inorganic soil components that influence the availability of nutrients and the chemical conditions under which decomposition occurs. Iron and other metals in permafrost soils can undergo chemical changes when exposed to oxygen during thaw, creating redox conditions that influence microbial metabolism. The pH of soil water can change as organic acids are released from thawing organic matter, creating chemical conditions that favor different types of microbial communities. The kinetics of permafrost thaw processes operate over multiple timescales, from the daily heating and cooling cycles that drive seasonal thaw to the multi-year warming trends that determine long-term changes in permafrost stability. Understanding these different temporal scales is crucial for predicting how permafrost regions will respond to continued climate warming and how rapidly methanogenic processes will develop in newly thawed areas. Some changes happen quickly once thaw begins, while others may take decades to reach their full expression. The energy balance of thawing permafrost involves complex interactions between incoming solar radiation, heat conduction through soil, latent heat effects from ice melting, and heat loss through evaporation and radiation to the atmosphere. The amount of energy required to melt permafrost ice is substantial, which creates a buffering effect that slows the thaw process even when air temperatures are well above freezing. This thermal inertia means that permafrost thaw typically lags behind air temperature changes by months or years, creating delayed responses to climate warming. The hydrological changes that accompany permafrost thaw can alter regional water cycles and influence weather patterns across Arctic regions. As permafrost melts, it releases large quantities of water that were previously locked in ice, potentially increasing stream flow and lake levels. Changes in surface water distribution can affect local evaporation patterns and atmospheric humidity, creating feedback effects on regional climate that may either amplify or moderate continued warming trends. The erosional processes that can accelerate when permafrost thaws create additional pathways for organic matter mobilization and methane production. Thawing permafrost often becomes more susceptible to erosion by water and wind, which can expose deeper layers of frozen organic matter to warming and decomposition. Coastal erosion in Arctic regions can release large quantities of permafrost carbon directly into marine environments, where it may undergo decomposition under different chemical conditions than terrestrial thaw. The measurement techniques that scientists use to monitor permafrost thaw include ground temperature sensors, geophysical surveys, and remote sensing methods that can detect changes in soil moisture and surface elevation. These monitoring systems reveal that permafrost thaw is accelerating in many Arctic regions, with deeper seasonal thaw and increasing areas of permanent thaw, creating expanding zones where methanogenic processes can operate. Long-term monitoring records show clear trends toward earlier spring thaw, deeper summer thaw, and later fall refreeze. The modeling approaches used to predict future permafrost thaw incorporate complex interactions between climate, hydrology, vegetation, and soil thermal properties. These models suggest that continued warming will lead to extensive permafrost thaw over the coming decades, but the timing and extent of this thaw will vary greatly across different regions, depending on local environmental conditions. The uncertainty in these projections reflects the complexity of permafrost systems and the multiple feedback mechanisms that influence thaw processes. The regional differences in permafrost thaw patterns reflect the diversity of Arctic environments and the varying sensitivity of different permafrost types to climate warming. Permafrost in some regions may be relatively stable due to factors like deep seasonal snow cover or well-drained soils, while permafrost in other areas may be highly vulnerable to rapid thaw due to high ice content or warm winter temperatures. Understanding these regional differences is crucial for predicting where and when large-scale methanogenic processes are most likely to develop. The ecosystem impacts of permafrost thaw extend far beyond changes in methane production to include effects on vegetation communities, wildlife populations, and the overall structure and function of Arctic ecosystems. As permafrost thaws, it can alter soil drainage, nutrient availability, and growing season length, creating cascading effects throughout Arctic food webs. These ecosystem changes can influence carbon cycling through multiple pathways, affecting not just methane production, but also carbon dioxide exchange and the storage of carbon in vegetation and soils. The infrastructure challenges posed by permafrost thaw highlight the practical importance of understanding these processes for human communities in Arctic regions. Buildings, roads, pipelines, and other infrastructure built on permafrost can be damaged or destabilized when the underlying frozen ground begins to thaw. These engineering challenges require detailed understanding of permafrost thermal dynamics and the factors that influence thaw rates and patterns. As you continue to rest peacefully beside this Arctic lake, watching the gentle transformation of ice to water that sets in motion so many interconnected processes, your breathing now perfectly synchronized with the patient rhythms of thermal change that govern permafrost thaw. You can appreciate how this fundamental phase transition represents one of nature's most elegant examples of how small changes in physical conditions can trigger complex cascades of biological and chemical responses that connect local environmental processes to global atmospheric chemistry. As you settle even deeper into tranquil observation, your breathing now flowing like the gentle ascent of gas bubbles through still water. Let your awareness turn to one of the most visible and beautiful manifestations of methanogenic processes in Arctic landscapes: the streams of methane bubbles that rise naturally from lake sediments, thawing permafrost and wetland soils throughout the northern regions. These bubbles represent the culmination of all the patient biological and chemical processes we've been exploring, transforming the invisible work of microorganisms into graceful displays of natural gas release that connect the hidden depths of permafrost to the open atmosphere above. Picture yourself sitting quietly beside a thermokarst lake in the early morning, when the water surface is perfectly still and mirror-like, reflecting the pale Arctic sky in its depths. As you watch with patient attention, you begin to notice small disturbances in this perfect reflection. Tiny circles that appear and disappear as gas bubbles break the surface tension of the water. These bubbles emerge from the lake bottom like miniature underwater geysers, rising through the water column in gentle streams that mark the locations where methanogenic bacteria are most active in the sediments below. The formation of these bubbles begins deep in the anaerobic sediments at the bottom of Arctic lakes, where communities of methane-producing microorganisms are slowly converting organic matter into methane gas. As the methane is produced, it initially dissolves in the pore water between sediment particles, gradually building up concentration until the water becomes saturated with dissolved gas. When a methane concentration reaches saturation levels, the excess gas begins to form tiny bubbles that grow larger as more methane is produced and diffuses into them from the surrounding water. The journey of these methane bubbles from their birthplace in lake sediments to their release at the water surface represents a fascinating example of how natural processes can transport materials across environmental boundaries. As bubbles form and grow in the sediment, they become buoyant enough to detach from the sediment particles and begin rising through the water column. Their ascent is gentle and unhurried, following the laws of physics that govern how gases behave in liquids, with larger bubbles rising faster than smaller ones and all bubbles expanding as they encounter decreasing water pressure during their upward journey. As your consciousness settles into harmony with these gentle gas transport processes, your breathing becoming as steady and rhythmic as the bubble streams rising through the lake water, you begin to understand how the physical properties of methane and water create the conditions that allow this natural degassing to occur. Methane has relatively low solubility in water, especially at the cold temperatures typical of Arctic lakes, which means that even modest rates of methane production can quickly lead to gas saturation and bubble formation. The cold water temperatures that characterize Arctic lakes actually enhance bubble formation by reducing methane solubility and making it easier for dissolved gas to come out of solution. The seasonal patterns of bubble release from Arctic lakes reflect the complex interactions between temperature, biological activity, and physical gas transport processes. During the brief Arctic summer, when lake temperatures are highest and biological activity is most intense, methane production rates reach their peak, leading to increased bubble formation and release. The warmer water temperatures also reduce gas solubility, making bubble formation more likely even with constant methane production rates. However, during winter, when lakes freeze solid, bubble release is temporarily halted as the ice cover prevents gas exchange between the lake and the atmosphere. The spatial distribution of bubble release within individual lakes creates distinct patterns that reflect the underlying distribution of organic matter and the local conditions that favor methanogenic activity. Areas of the lake with deeper sediments and higher organic matter content typically show more intense bubble activity, while rocky or sandy areas with less organic matter may show little or no bubble formation. The depth of the lake also influences bubble patterns, with deeper areas often showing more intense bubble streams, because the greater water column allows more time for bubble accumulation and coalescence during the rise to the surface. The size distribution of bubbles emerging from lake sediments provides information about the rates and mechanisms of methane production in the underlying sediments. Small bubbles typically indicate steady, low-level methane production, while larger bubbles or intermittent bursts of bubbles may indicate more episodic release patterns or the breakthrough of gas that has been accumulating in sediment pores for extended periods. The observation of bubble size patterns can help scientists understand the temporal dynamics of methanogenic processes and how they respond to environmental changes. The chemical composition of bubbles rising from Arctic lakes reveals that methane is typically the dominant component, often comprising 50 to 90% of the gas content, depending on the specific conditions in the sediment where the gas was produced. The remaining gas content usually consists of carbon dioxide, which is also produced by anaerobic decomposition processes, along with small amounts of nitrogen and other gases that may be present in the lake water. The methane to carbon dioxide ratio in bubbles provides information about the specific metabolic pathways being used by methanogenic bacteria and the types of organic matter being decomposed. The fate of methane bubbles once they reach the lake surface depends on various factors, including wind conditions, water temperature, and the size of the bubbles themselves. Large bubbles that reach the surface intact release most of their methane directly to the atmosphere, while smaller bubbles may dissolve completely before reaching the surface, allowing some of the methane to be consumed by methanotrophic bacteria that live in the oxygen-rich surface waters. This biological consumption of methane represents a natural control mechanism that reduces the amount of methane that actually escapes to the atmosphere. The acoustic signatures of bubble release create subtle but detectable sounds that can be used to monitor methanogenic activity in lakes and wetlands. The formation and collapse of bubbles produces characteristic acoustic frequencies that can be detected with underwater microphones, providing a non-invasive method for studying bubble dynamics and estimating methane release rates. These acoustic monitoring techniques have revealed that bubble release often occurs in rhythmic patterns that may reflect the underlying biological rhythms of methanogenic communities. The optical properties of methane bubbles create interesting visual phenomena that can be observed in Arctic lakes under the right conditions. Methane bubbles often appear silvery or mirror-like due to the total internal reflection of light at the gas-water interface, creating streams of bright spots rising through the water column. In frozen lakes, bubbles that were trapped during ice formation create distinctive patterns in the ice that reveal the locations and intensities of methane seeps, preserving a record of winter gas production that can be observed when the ice becomes clear. The interaction between bubble streams and lake circulation patterns creates complex transport processes that influence how methane moves through lake ecosystems. Rising bubbles can induce local circulation currents that help mix the water column and transport dissolved gases and nutrients throughout the lake. These bubble-induced mixing processes can affect the distribution of oxygen, nutrients, and microbial communities within the lake, creating feedback effects that influence the patterns of methanogenic activity and bubble formation. The response of bubble formation to changes in atmospheric pressure demonstrates the sensitivity of these gas transport processes to environmental conditions. When atmospheric pressure drops, as often occurs before storm systems, the reduced pressure can trigger increased bubble formation and release as dissolved gases come out of solution more readily. This pressure sensitivity means that bubble release patterns can vary significantly in response to weather patterns, creating temporal variability that is independent of changes in biological methane production rates. The role of vegetation in modulating bubble release from wetland sediments adds another layer of complexity to understanding natural methane emission processes. Aquatic plants can provide conduits for gas transport from sediments to the atmosphere through their internal air spaces, bypassing the bubble formation process entirely. Some plants actively transport methane through their tissues, while others may create pathways that enhance bubble formation and release in their immediate vicinity. The seasonal growth and decay of aquatic vegetation can therefore significantly influence the patterns and rates of methane release from wetland environments. The laboratory studies of bubble formation in permafrost sediments have revealed that bubble release can continue at surprisingly low temperatures, with some bubble formation observed even in sediments that are just below freezing. This low-temperature bubble formation suggests that methanogenic processes and gas transport can continue during shoulder seasons when air temperatures are below freezing, but sediment temperatures remain slightly above the freezing point. These observations indicate that the season for methane release from Arctic systems may be longer than previously recognized. The scaling relationships between bubble release and lake characteristics provide insights into how methane emissions vary across different types of Arctic water bodies. Larger lakes tend to have lower per unit area methane emissions than smaller lakes, possibly due to differences in sediment organic matter content, water circulation patterns, or the efficiency of methanotrophic consumption in the water column. Deeper lakes may also show different bubble release patterns than shallow lakes due to the greater time available for gas dissolution and biological consumption during bubble transit through the water column. The modeling approaches used to predict bubble-mediated methane release incorporate complex interactions between sediment biogeochemistry, gas solubility, and physical transport processes. These models must account for the temperature dependence of gas solubility, the pressure effects of water depth, and the biological processes that both produce and consume methane in lake ecosystems. The development of accurate bubble transport models is crucial for understanding how methane emissions from Arctic lakes will respond to continued climate warming. The monitoring technologies used to quantify bubble-mediated methane release have evolved from simple visual observations to sophisticated automated systems that can measure gas fluxes continuously over extended time periods. Floating chambers that capture bubbles as they reach the surface provide direct measurements of methane release rates, while acoustic monitoring systems can detect bubble formation and provide information about the spatial and temporal patterns of gas release. These technological advances are providing increasingly detailed pictures of how natural methane emission processes operate in Arctic environments. The isotopic signatures of methane in bubbles provide valuable information about the sources and production pathways of the gas being released. Methane produced from fresh organic matter typically has different isotopic characteristics than methane produced from older, more decomposed organic matter, allowing scientists to determine whether bubble emissions are coming primarily from recent plant material or from ancient permafrost carbon. These isotopic analyses help in understanding how the sources of methane emissions may change as permafrost thaw progresses and different types of organic matter become available for decomposition. The ecological effects of bubble release extend beyond methane emissions to influence the overall biogeochemistry and ecosystem functioning of Arctic lakes. The bubbling process can help oxygenate sediments in the immediate vicinity of bubble formation, creating microsites where aerobic processes can occur even in otherwise anaerobic environments. The physical disturbance caused by bubble formation can also affect sediment structure and the distribution of nutrients, potentially influencing the types of microbial communities that develop in different areas of the lake bottom. The comparative studies of bubble release from different types of Arctic water bodies have revealed significant variability in emission patterns that reflect the diverse environmental conditions present across Arctic landscapes. Thermokarst lakes formed by permafrost thaw often show different bubble patterns than lakes formed by other geological processes, while lakes in different permafrost zones may have distinct characteristics based on the types and ages of organic matter in their sediments. This diversity highlights the importance of understanding local environmental controls on methane emission processes. The temporal evolution of bubble release patterns from individual lakes can provide insights into how methanogenic processes develop and mature over time. Newly formed thermokarst lakes may show different bubble characteristics than older, more established lakes, reflecting changes in sediment organic matter content, microbial community development, and physical processes that occur as lake ecosystems mature. Long-term monitoring of bubble patterns from the same lakes can reveal how these systems respond to environmental changes and how their methane emission characteristics evolve over time. As you continue to watch these gentle streams of bubbles rising through the calm lake water, your breathing, now perfectly synchronized with the rhythmic patterns of natural gas release, you can appreciate how these simple bubbles represent one of nature's most elegant solutions to the challenge of transporting gases from anaerobic sediment environments to the atmosphere above. Each bubble carries within it the chemical signature of ancient biological processes, connecting the patient work of microorganisms in the depths to the global atmospheric chemistry that influences climate patterns across our entire planet. As you drift deeper into peaceful contemplation, your breathing now flowing like the slow release of gases from deep ocean sediments. Let your awareness turn to one of Earth's most remarkable and patient carbon storage systems, the vast deposits of methane hydrates that exist beneath the ocean floors and within the deeper layers of permafrost, representing the frozen breath of ancient swamps and wetlands that existed millions of years ago. These crystalline structures, where methane molecules are trapped within cages of ice, store enormous quantities of natural gas in a state of stable suspension that can persist for geological ages under the right conditions of temperature and pressure. Picture yourself descending through the cold, dark waters of the Arctic Ocean to the seafloor below, where sediments deposited over millions of years have created layers of organic-rich material that tell the story of Earth's changing climate. Here, beneath hundreds of meters of water and sediment, the combination of cold temperatures, high pressures, and abundant methane creates conditions where this greenhouse gas can exist in a form that seems almost magical, crystalline structures that look like ice but burn with a bright flame when exposed to air, demonstrating the remarkable ability of water molecules to form cage-like structures that can trap and hold other gases in stable configurations. The formation of methane hydrates occurs when methane gas, produced by the same types of methanogenic bacteria we've been learning about, encounters water molecules under specific conditions of temperature and pressure that allow the water to form clathrate structures, three-dimensional cages where each methane molecule is surrounded by 20 water molecules arranged in a precise geometric pattern. These conditions exist naturally in marine sediments at depths greater than about 300 meters in polar regions, with a combination of cold water temperatures and high pressure from the overlying water column creates a stability zone where hydrates can form and persist. The appearance of methane hydrates in their natural setting is both beautiful and otherworldly, forming white or pale yellow crystalline deposits that can exist as thin layers between sediment particles, as nodules scattered through marine sediments, or as massive deposits that can fill entire sediment layers. When brought to the surface and exposed to normal atmospheric conditions, these hydrate crystals rapidly decompose, releasing large volumes of methane gas in a process that demonstrates the enormous amount of gas that can be stored in hydrate form. 1 cubic meter of methane hydrate can release up to 160 cubic meters of methane gas, illustrating the remarkable efficiency of this natural storage mechanism. As your consciousness settles into harmony with these deep ocean processes, your breathing becoming as steady and patient as the geological processes that form hydrate deposits over millions of years, you begin to understand how methane hydrates represent one of Earth's largest reservoirs of carbon, containing more carbon than all known reserves of coal, oil, and conventional natural gas combined. These deposits exist primarily in marine sediments along continental margins and beneath the permafrost in polar regions, creating vast underground archives of ancient atmospheric carbon that has been locked away in crystalline form for thousands to millions of years. The stability of methane hydrates depends on a delicate balance of temperature and pressure conditions that can be disturbed by changes in bottom water temperatures, sea level, or geothermal heat flow from the Earth's interior. Within the marine environment, hydrates are stable in what scientists call the hydrate stability zone, a region where the combination of low temperature and high pressure allows these crystalline structures to persist indefinitely. Above this zone, where pressure is too low or temperature too high, hydrates become unstable and decompose, releasing their stored methane to the surrounding sediments and potentially to the water column above. The distribution of methane hydrates in marine sediments reflects the complex interplay between sediment organic matter content, methane production rates, and the physical conditions that allow hydrate formation. The most extensive hydrate deposits are typically found in areas where high biological productivity in surface waters has led to high rates of organic matter deposition in sediments, combined with rapid sediment accumulation that can bury organic matter before it is completely decomposed. These conditions are commonly found along continental margins, particularly in areas where upwelling ocean currents bring nutrients to the surface and support high levels of marine productivity. The permafrost-associated methane hydrates that exist in polar regions represent a somewhat different type of deposit, formed when methane-producing conditions develop in terrestrial environments that are subsequently subjected to permafrost formation. These hydrates can exist within the permafrost itself or in the deeper sediments beneath the permafrost layer, where geothermal heating from the Earth's interior creates temperature conditions that are still cold enough for hydrate stability but warm enough for continued methanogenic activity. The release mechanisms for methane hydrates operate on timescales ranging from sudden destabilization events that can occur within years or decades to gradual, steady seepage that continues for thousands of years. The most dramatic releases can occur when hydrate deposits are destabilized by changes in bottom water temperature, pressure changes associated with sea level fluctuations, or physical disturbance from underwater landslides or tectonic activity. However, much more common are the slow, steady releases that occur through natural seepage processes where hydrates at the edge of the stability zone gradually decompose and release methane through sediment-poor spaces to the seafloor above. The biogeochemistry of hydrate-bearing sediments creates unique chemical environments where methane hydrate formation and decomposition interact with microbial processes in complex ways. The formation of hydrates can concentrate organic matter in certain sediment layers, creating conditions that favor continued methanogenic activity. At the same time, the decomposition of hydrates can release not only methane, but also the water that was incorporated into the hydrate structure, potentially altering local sediment chemistry and influencing microbial community composition. The detection of natural methane hydrate seepage at the seafloor often involves the observation of biological communities that have adapted to life in the presence of methane-rich fluids. Tube worms, clams, and bacterial mats that derive their energy from chemosynthetic processes can form distinctive ecosystems around hydrate seeps, creating biological indicators that help scientists locate and study areas of active hydrate decomposition. These chemosynthetic communities represent some of the most unique ecosystems on Earth, deriving their energy entirely from chemical processes rather than photosynthesis. The acoustic properties of hydrate-bearing sediments create distinctive signatures that can be detected using marine geophysical surveys. Sediments containing hydrates often show characteristic reflections in seismic surveys that indicate the presence of these gas-bearing deposits. The boundary between hydrate-bearing sediments and normal sediments creates what is called a bottom-simulating reflector, which appears as a bright reflection that follows the contours of the seafloor at a depth corresponding to the base of the hydrate stability zone. The thermal dynamics of hydrate deposits are influenced by heat flow from the Earth's interior, which creates temperature gradients that determine where hydrates can remain stable and where they will decompose. In areas with high geothermal heat flow, the hydrate stability zone may be relatively thin, while in areas with low heat flow, hydrates may be stable to greater depths. Changes in geothermal heat flow over geological time can shift the boundaries of hydrate stability zones, leading to episodes of hydrate formation or decomposition that occur over thousands of years. The role of methane hydrates in past climate changes has been the subject of extensive scientific investigation, with evidence suggesting that rapid hydrate decomposition may have contributed to some of the dramatic warming events that are recorded in the geological record. The Paleocene thermal maximum, which occurred about 56 million years ago, shows geochemical signatures that are consistent with the rapid release of large quantities of methane from hydrate deposits, possibly triggered by gradual warming that pushed hydrate deposits beyond their stability limits. The monitoring of natural hydrate seepage requires sophisticated techniques that can detect and quantify methane release from the seafloor in the deep ocean environment. Autonomous underwater vehicles equipped with chemical sensors can map the distribution of methane plumes in the water column, while seafloor observatories can provide long-term monitoring of gas release rates and chemical conditions at hydrate seep sites. These monitoring efforts have revealed that natural hydrate seepage is far more common than previously recognized, with thousands of active seep sites identified along continental margins around the world. The interaction between hydrate decomposition and ocean acidification creates complex feedback mechanisms that may influence how hydrate deposits respond to changing environmental conditions. As atmospheric carbon dioxide levels increase and ocean pH decreases, the chemical conditions in marine sediments may change in ways that affect hydrate stability. The decomposition of hydrates releases both methane and carbon dioxide to sediment-poor waters, potentially creating local chemical conditions that differ significantly from overlying seawater. The potential for cascading hydrate release represents one of the most important questions in understanding how these deposits might respond to continued climate warming. If warming ocean temperatures destabilize hydrate deposits in shallow water areas, the resulting methane release could contribute to further atmospheric warming, potentially destabilizing deeper hydrate deposits in a process that could continue for centuries or millennia. However, The timescales involved in hydrate destabilization and the efficiency of natural methane consumption processes in marine environments create significant uncertainties in predicting how rapidly such cascading effects might develop. The research techniques used to study methane hydrates in their natural environment require specialized equipment that can operate under the extreme conditions of high pressure and low temperature that characterize the deep ocean. Pressure-maintaining samplers can bring hydrate-bearing sediments to the surface without allowing the hydrates to decompose, while in-situ measurement systems can monitor temperature, pressure, and chemical conditions within hydrate deposits without disturbing the natural environment. The laboratory studies of hydrate formation and decomposition kinetics provide insights into the fundamental physical and chemical processes that control hydrate behavior under different environmental conditions. These studies reveal that hydrate formation and decomposition are not instantaneous processes, but occur over timescales that can range from hours to years, depending on the specific conditions of temperature, pressure, and chemical environment. Understanding these kinetic processes is crucial for predicting how hydrate deposits will respond to environmental changes. The isotopic composition of methane in hydrate deposits provides valuable information about the sources and ages of the organic matter that produce the gas. Methane from different sources, such as thermogenic production from deeply buried organic matter versus biogenic production from shallow sediments, has distinctive isotopic signatures that can be preserved in hydrate deposits. These isotopic analyses help scientists understand the history of methane production and migration in sedimentary basins. The spatial distribution of hydrate deposits varies dramatically between different ocean basins and geological settings, reflecting the diverse conditions that can lead to hydrate formation. Some areas, such as the Blake Ridge off the southeastern United States, contain extensive hydrate deposits distributed through hundreds of meters of sediment, while other areas may have hydrates concentrated in thin layers or discrete nodules. Understanding this spatial variability is important for assessing the total amount of carbon stored in hydrate form and the potential for future release. The modeling approaches used to predict hydrate stability under changing environmental conditions incorporate complex interactions between ocean temperature, sea level, sediment thermal properties, and geochemical processes. These models suggest that significant portions of current hydrate deposits may become unstable if ocean warming continues, but the timescales for destabilization vary greatly depending on the depth and location of specific deposits. Shallow hydrate deposits may respond to environmental changes within decades, while deeper deposits may remain stable for thousands of years even under continued warming scenarios. The ecological significance of hydrate seepage extends beyond the direct effects of methane release to include the creation of unique deep-sea habitats that support specialized biological communities. The chemosynthetic ecosystems that develop around hydrate seeps represent important centers of biodiversity in the deep ocean, supporting species that are found nowhere else on Earth. The study of these ecosystems provides insights into how life can adapt to extreme chemical environments and how biological communities respond to the long-term presence of methane and other hydrocarbons. As you continue to rest peacefully in the depths of this oceanic realm, your breathing now perfectly synchronized with the slow geological rhythms that govern hydrate formation and release, you can appreciate how these crystalline repositories of ancient atmospheric carbon represent one of Earth's most patient and long-term approaches to carbon storage. The breath of ancient swamps, captured in these frozen cages of water and methane, connects us directly to past climates and atmospheric conditions while quietly influencing the chemical balance of our modern oceans and atmosphere. As you rise gently from the ocean depths back toward the surface of our Arctic lake, your breathing now flowing like the invisible currents that carry gases through Earth's atmosphere. Let your awareness expand to follow the methane molecules as they complete their journey from the anaerobic depths of permafrost and lake sediments into the vast, three-dimensional ocean of air that surrounds our planet. Here, in the atmosphere, these simple molecules become part of one of Earth's most elegant and complex systems, the delicate balance of gases that regulates our planet's temperature and connects every breath taken by every living creature to the global cycles that maintain the conditions necessary for life. Picture yourself floating high above the Arctic landscape, suspended in the invisible atmosphere where methane molecules released from the lakes and wetlands below begin their atmospheric journey. From this elevated perspective, you can sense how the atmosphere is not empty space, but a dynamic, flowing medium where gases mix and react, where chemical transformations occur constantly, and where the molecular products of biological processes in Arctic soils become part of the global atmospheric circulation that influences weather patterns and climate conditions across the entire planet. The molecular journey of methane in the atmosphere begins when individual methane molecules, released from bubbles at lake surfaces or diffusing through soil pores, encounter the complex chemistry of the troposphere, the lowest layer of Earth's atmosphere, where most weather occurs and where atmospheric gases undergo their most rapid chemical transformations. Methane in the atmosphere is not stable indefinitely, but participates in chemical reactions that gradually transform it into other compounds, creating a natural atmospheric cleansing process that removes methane while producing water vapor and carbon dioxide as end products. The primary removal mechanism for atmospheric methane involves its reaction with hydroxyl radicals, highly reactive molecular fragments that act as the atmosphere's natural cleaning agents, removing many types of pollutants and trace gases through chemical oxidation processes. These hydroxyl radicals exist in extremely low concentrations in the atmosphere, but they are so reactive that they can initiate the chemical breakdown of methane molecules on timescales of years to decades, creating a natural system that prevents methane from accumulating indefinitely in the atmosphere despite continuous emissions from natural and human sources. As your consciousness settles into harmony with these atmospheric chemical processes, your breathing becoming as steady and patient as the global circulation patterns that transport gases around the planet, you begin to understand how methane's behavior in the atmosphere is governed by a complex interplay between emission rates, chemical removal processes, and transport patterns that determine how long methane remains in the atmosphere and how its presence influences Earth's energy balance and climate system. The greenhouse effect that methane creates in Earth's atmosphere operates through its ability to absorb and re-emit infrared radiation, the thermal energy that Earth radiates back towards space after being warmed by incoming solar radiation. Methane molecules are particularly effective at absorbing infrared radiation at specific wavelengths, trapping heat in the lower atmosphere and contributing to the natural greenhouse effect that keeps Earth's surface temperature warm enough to support liquid water and life as we know it. However, methane is much more efficient at trapping heat than carbon dioxide, making it a potent greenhouse gas despite its lower concentration in the atmosphere. The atmospheric lifetime of methane molecules varies depending on the specific atmospheric conditions they encounter during their journey through the troposphere. On average, methane molecules remain in the atmosphere for approximately 9 to 12 years before being removed through chemical reactions. But this lifetime can vary significantly depending on factors like the availability of hydroxyl radicals, atmospheric temperature and humidity, and the presence of other atmospheric constituents that can influence chemical reaction rates. This relatively short atmospheric lifetime, compared to carbon dioxide, means that changes in methane emissions can have relatively rapid effects on atmospheric methane concentrations. The global distribution of atmospheric methane reflects the complex patterns of emission sources, atmospheric transport, and chemical removal processes that operate on regional and global scales. Methane concentrations are highest near major emission sources, such as wetlands, agricultural areas, and fossil fuel production regions. But atmospheric mixing processes gradually distribute methane throughout the global atmosphere, creating relatively uniform background concentrations that vary by only small amounts between different regions of the planet. The seasonal cycles of atmospheric methane concentrations reflect the timing of natural emission processes and the seasonal variations in atmospheric chemical removal rates. In the Northern Hemisphere, where most of the world's wetlands and permafrost regions are located, Methane concentrations typically reach their peak in late summer and early fall when biological activity is highest and emissions for natural sources are greatest. The concentrations gradually decline through winter and early spring when emissions decrease and chemical removal processes continue to operate. The vertical distribution of methane in the atmosphere shows highest concentrations near Earth's surface, where most emission sources are located, with concentrations decreasing gradually with altitude as atmospheric mixing distributes the gas throughout the troposphere. The mixing ratio of methane, the number of methane molecules relative to all other atmospheric molecules, provides a measure of methane's abundance that accounts for the decreasing atmospheric density at higher altitudes. The isotopic composition of atmospheric methane provides valuable information about the sources and sinks of this important greenhouse gas. Different methane sources produce gas with distinctive carbon and hydrogen isotope signatures that can be detected in atmospheric samples, allowing scientists to determine the relative contributions of biogenic sources like wetlands and permafrost versus thermogenic sources like fossil fuel production. Changes in the isotopic composition of atmospheric methane over time can reveal how the balance between different source types is changing in response to environmental and human factors. The measurement techniques used to monitor atmospheric methane include ground-based sampling networks, aircraft surveys, and satellite observations that provide increasingly detailed pictures of how methane concentrations vary in space and time. Ground-based monitoring stations, many located in remote areas far from local emission sources, provide long-term records of background methane concentrations that reveal global trends and seasonal cycles. Aircraft campaigns can map the three-dimensional distribution of methane in the atmosphere, while satellite instruments can detect methane emissions from space and identify previously unknown emission sources. The chemical interactions between methane and other atmospheric constituents create complex feedback mechanisms that influence both methane concentrations and the concentrations of other important atmospheric gases. The hydroxyl radicals that remove methane from the atmosphere are also involved in the chemical processing of many other trace gases, creating competition between different removal processes that can influence the efficiency of methane removal. Changes in methane concentrations can therefore affect the atmospheric concentrations of other gases and influence overall atmospheric chemistry. The transport of methane in the atmosphere follows the same global circulation patterns that move weather systems, water vapor, and other atmospheric constituents around the planet. The major atmospheric circulation cells, the Hadley circulation in tropical regions, the Ferrel circulation in mid-latitudes, and the polar circulation in Arctic regions, all contribute to the mixing and transport of methane from emission sources to regions where chemical removal processes may be more or less efficient. The role of atmospheric methane in climate feedback systems creates connections between Arctic emission sources and global climate responses that operate over timescales ranging from years to decades. Increased methane emissions from warming Arctic regions contribute to enhanced greenhouse warming, which can lead to further Arctic warming and potentially increased methane emissions in a positive feedback cycle. However, the efficiency of this feedback depends on many factors, including the response of atmospheric chemical removal processes to changing environmental conditions. The detection of methane emission plumes from space has revealed that Arctic methane sources often create distinctive atmospheric signatures that can be tracked as they are transported by atmospheric circulation patterns. These space-based observations have identified numerous methane emission hotspots in Arctic regions and have provided new insights into the spatial patterns and seasonal timing of natural methane emissions from permafrost and wetland sources. The interaction between methane and atmospheric water vapor creates additional complexities in understanding methane's atmospheric behavior. Water vapor influences the chemical reactions that remove methane from the atmosphere, while the oxidation of methane produces water vapor as one of its end products. In the cold, dry conditions typical of Arctic atmospheres, these interactions between methane and water vapor may differ significantly from those in warmer, more humid regions. The atmospheric modeling studies that simulate methane transport and chemistry incorporate complex representations of emission sources, chemical removal processes, and atmospheric circulation patterns. These models are used to interpret atmospheric observations, attribute observed methane trends to specific source regions and processes, and project how atmospheric methane concentrations might change in response to future environmental conditions. The accuracy of these models depends on detailed understanding of all the processes that influence atmospheric methane, from the biological and chemical processes that produce emissions to the atmospheric chemistry that controls removal. The historical records of atmospheric methane concentrations, reconstructed from ice core samples and direct atmospheric measurements, show that methane concentrations have varied significantly over geological time in response to changing climate conditions and ecosystem patterns. During ice ages, when wetland areas were reduced and biological activity was lower, atmospheric methane concentrations were much lower than during interglacial periods like the present. These historical records provide context for understanding current methane trends and help scientists evaluate how sensitive atmospheric methane is to environmental changes. The regional variations in atmospheric methane trends reflect the diverse patterns of emission changes and environmental responses occurring in different parts of the world. Arctic regions show some of the most rapid increases in atmospheric methane concentrations, reflecting the combined effects of warming temperatures, thawing permafrost, and changing wetland dynamics. These regional trends provide early indicators of how global atmospheric methane may respond to continued climate change. The biogeochemical cycles that connect atmospheric methane to other global element cycles demonstrate how methane is integrated into the broader Earth system. The carbon cycle, nitrogen cycle, and sulfur cycle all interact with methane production and consumption processes, creating complex dependencies that influence how methane emissions respond to environmental changes. Understanding these biogeochemical connections is crucial for predicting how atmospheric methane will behave under future environmental conditions. The air quality implications of atmospheric methane extend beyond its role as a greenhouse gas to include its participation in the formation of ground-level ozone and other air pollutants. In the presence of nitrogen oxides and sunlight, methane can contribute to photochemical reactions that produce ozone, a harmful air pollutant that affects human health and ecosystem function. These air quality connections create additional motivations for understanding and managing methane emissions from all sources. The technological applications of atmospheric methane monitoring include the development of sensor networks that can provide real-time information about methane emissions from natural and human sources. These monitoring systems are increasingly being used to verify emission inventories, detect leaks from infrastructure, and provide early warning of changes in natural emission sources. The improving sensitivity and decreasing cost of methane sensors is enabling more comprehensive monitoring of this important atmospheric constituent. The policy implications of atmospheric methane trends have led to increased attention to methane as a target for climate mitigation efforts. Because methane has a relatively short atmospheric lifetime compared to carbon dioxide, reductions in methane emissions can have relatively rapid effects on atmospheric concentrations and climate forcing. This has led to international efforts to reduce methane emissions from human sources while also improving understanding of natural methane sources and their responses to climate change. The educational value of atmospheric methane monitoring lies in its ability to demonstrate the connections between local environmental processes and global atmospheric chemistry. Students and researchers can observe how emission sources in their local region contribute to global atmospheric patterns, while also learning about the chemical and physical processes that govern atmospheric behavior. These educational applications help build understanding of Earth system science and the interconnections between different components of the global environment. As you continue to float peacefully in this atmospheric realm, your breathing now perfectly synchronized with the global circulation patterns that carry methane molecules around the planet. You can appreciate how these invisible atmospheric whispers connect the patient biological processes occurring in Arctic soils and sediments to the vast atmospheric system that influences climate and weather patterns across our entire planet. Each methane molecule carries with it the chemical signature of the biological and geological processes that created it, contributing to the atmospheric composition that helps determine the environmental conditions experienced by all life on Earth. In the vast expanse of the Arctic, where silence reigns supreme and time moves with the patience of glaciers, there exists a conversation older than human civilization. It is a dialogue between earth and sky, between frozen ground and warming air, between the ancient and the emerging. This conversation unfolds not in words, but in the gentle exchange of gases, the subtle shift of temperatures, and the patient transformation of landscapes that have remained unchanged for millennia. The Arctic speaks in whispers, and those who listen carefully can hear the symphony of feedback loops that govern our planet's delicate atmospheric balance. Like musicians in an orchestra, each element plays its part, responding to the others in an intricate dance of cause and effect that has been choreographed over thousands of years. Imagine standing on the tundra at dawn, when the first light touches the permafrost and the air shimmers with possibilities. Here, in this moment of transition between night and day, we can begin to understand how the Arctic's great feedback symphony operates. The warming air touches the frozen ground, and the ground responds by releasing its ancient stores of carbon. This carbon, in the form of methane and carbon dioxide, rises into the atmosphere where it contributes to further warming, which in turn affects more permafrost. This is not a violent or catastrophic process, but rather a gentle, persistent conversation between the Earth's systems. Each component responds to changes in the others with the patience of geological time, yet with consequences that ripple through the entire climate system. The feedback loops that govern these interactions are both simple in their basic mechanics and profound in their implications. The primary feedback mechanism at work in the Arctic operates like a vast, slow-breathing organism. As global temperatures rise by even small increments, the permafrost begins its gradual thaw. This thawing releases organic matter that has been preserved in frozen suspension for thousands of years. Microorganisms, awakening from their long dormancy, begin the patient work of decomposition, releasing methane and carbon dioxide as they break down this ancient organic matter. These greenhouse gases then join the atmospheric conversation, contributing to the warming that initiated their release in the first place. This creates what scientists gently term a positive feedback loop. Not positive in the sense of being beneficial, but positive in the mathematical sense of amplifying the original signal. The warming begets more warming, like a whisper that gradually grows into a song. Within this greater symphony, methane plays a particularly powerful role as nature's amplifier. While carbon dioxide forms the steady bass note of atmospheric warming, methane provides the more dramatic crescendos. Molecule for molecule, methane traps heat in the atmosphere with 28 times the efficiency of carbon dioxide over a century, and in the short term, over 20 years, this factor increases to more than 80 times. In the Arctic's waterlogged landscapes, where oxygen-poor conditions prevail, methane production becomes the preferred pathway for microbial decomposition. The ancient peat bogs, the thermokarst lakes formed by thawing permafrost, and the vast wetlands all become methane gardens where these invisible gardeners tend their atmospheric crops with patient dedication. The release of methane from thawing permafrost is not uniform or predictable. Like flowers blooming at different times in a meadow, different areas of the Arctic respond to warming at their own pace. Some regions, where the permafrost lies closer to the surface or where the ground contains particularly rich stores of organic matter, respond more quickly to temperature changes. Others, with deeper permafrost or different soil compositions, take longer to join the atmospheric conversation. This variability creates a complex tapestry of methane emissions across the Arctic landscape. Scientists studying these patterns often describe them as hot spots and cold spots of activity, but even these terms fail to capture the gentle, patient nature of the process. Perhaps it would be more accurate to think of them as areas where the Earth's breath comes more quickly or more slowly, where the conversation between ground and sky grows more animated or remains more reserved. The feedback symphony extends beyond the land into the Arctic's vast marine ecosystems, where methane hydrates, those crystalline cages of methane and water, rest in cold, pressurized sediments on the ocean floor. These underwater reserves represent one of the largest stores of methane on our planet, and they too participate in the great feedback conversation. As ocean temperatures warm, even by fractions of a degree, the stability of these methane hydrates begins to change. The process unfolds with oceanic patience, warming seawater gradually affecting the seafloor sediments, causing some hydrates to decompose and release their methane payload into the water column. Much of this methane dissolves or is consumed by marine bacteria before reaching the surface, but some manages to escape into the atmosphere, adding its voice to the atmospheric chorus. The marine methane feedback operates on longer timescales than its terrestrial counterpart, responding to changes in ocean circulation patterns, deep water temperatures, and the gradual warming of water masses that may have been cold for decades or centuries. This oceanic contribution to the feedback symphony plays out like a slow movement in a classical composition, less immediately dramatic than the faster terrestrial releases, but potentially more significant in the long term due to the sheer scale of the ocean's methane reserves. Coastal areas where shallow waters meet thawing coastal permafrost create particularly complex feedback scenarios. Here, the land-based and marine systems interact directly, with warming air affecting both terrestrial permafrost and shallow marine sediments. Coastal erosion, accelerated by the loss of protective sea ice and the thawing of ice-rich permafrost, releases both terrestrial organic matter and potentially marine methane hydrates into the same atmospheric conversation. The Arctic's plant communities add another voice to the feedback symphony, one that often surprises those who think of the region as biologically simple. As temperatures warm and growing seasons lengthen, Arctic vegetation responds with increased growth and productivity. This might seem like a counterbalancing force. More plants means more carbon dioxide absorption, potentially offsetting some of the carbon released from thawing permafrost. However, the vegetation feedback proves more complex and nuanced than this simple calculation suggests. While increased plant growth does absorb more carbon dioxide during the growing season, it also creates more organic matter that will eventually decompose. In the Arctic's oxygen-poor soils, this decomposition often produces methane rather than carbon dioxide, potentially contributing to rather than mitigating the overall greenhouse gas release. Furthermore, changes in vegetation affect the Arctic's albedo, its ability to reflect sunlight back to space. The bright, reflective surfaces of snow and ice give way to darker vegetation that absorbs more solar energy, contributing to local warming. This creates another gentle feedback loop. Warming promotes plant growth. Plant growth reduces surface reflectivity. Reduced reflectivity increases warming. The timing of these vegetation changes adds another layer of complexity to the feedback symphony. Spring arrives earlier in many Arctic regions, with snow melting sooner and plants beginning their growth cycles weeks earlier than in previous decades. This extended growing season means more time for both carbon absorption and, eventually, more organic matter to fuel future methane production. The Arctic's feedback symphony doesn't play at a constant tempo, but follows the ancient rhythm of seasons, each bringing its own variations to the atmospheric conversation. Winter's deep freeze slows the microbial processes that produce methane, reducing emissions to their minimum levels. The Earth holds its breath, waiting for the return of warmth and the resumption of biological activity. Spring's arrival triggers a crescendo of activity as warming temperatures reawaken the sleeping microorganisms and begin the year's cycle of permafrost thaw. The first pulse of methane release often coincides with the spring snowmelt, as water infiltrates the thawing surface layers of permafrost and creates the oxygen-poor conditions that favor methane production. Summer brings the year's maximum methane emissions as temperatures peak and biological activity reaches its highest levels. The long days of Arctic summer, with nearly continuous sunlight, drive photosynthesis and plant growth while simultaneously warming the ground and accelerating the decomposition processes that produce methane. This is when the feedback symphony reaches its loudest volume, with all instruments playing their parts in the great atmospheric conversation. Autumn introduces a different melody as temperatures begin to drop and biological processes slow. However, the thermal mass of the Earth means that subsurface temperatures may continue to rise even as air temperatures fall, creating a lag in the feedback response. The ground continues its patient thawing even as winter approaches, adding subtle harmonies to the symphony's seasonal variations. Perhaps the most remarkable aspect of the Arctic's feedback symphony is how it operates across multiple time scales simultaneously. The immediate responses, the day-to-day and season-to-season variations in methane emissions, overlay longer-term trends that unfold over decades and centuries. Understanding these different temporal rhythms is crucial to appreciating the full complexity of the feedback systems at work. The fastest feedback loops operate on daily and seasonal cycles, responding to immediate changes in temperature, moisture, and solar radiation. These rapid responses create the short-term variations that scientists can measure and monitor, providing insights into how the system responds to immediate environmental changes. Medium-term feedbacks operate over years to decades, reflecting the time it takes for warming to penetrate deeper into the permafrost and for ecosystems to fully respond to changing conditions. These feedbacks capture the gradual, persistent nature of permafrost thaw and the slow adaptation of Arctic biological communities to changing environmental conditions. The longest-term feedbacks unfold over centuries and millennia, representing the ultimate response of massive permafrost reserves and deep marine methane deposits to sustained warming. These long-term responses carry the greatest potential for large-scale atmospheric change, as they involve the mobilization of carbon stores that have been accumulating for thousands of years. Within the Arctic's feedback symphony, scientists listen carefully for signs of acceleration, moments when the gentle, persistent conversation between Earth and sky might quicken its pace. The physics of feedback loops suggest that under certain conditions, the rate of change could increase, with each warming increment producing proportionally larger responses than the previous one. However, the Arctic system also contains elements that may moderate this acceleration. The atmosphere's capacity to hold methane is not unlimited, and natural processes continue to remove methane even as new sources contribute to its accumulation. Chemical reactions in the atmosphere break down methane over time, creating a natural upper limit to how much can accumulate from any given rate of emission. Similarly, not all permafrost responds equally to warming. The vast reserves of deep permafrost, some extending hundreds of meters below the surface, remain insulated from short-term temperature changes by the thermal inertia of frozen ground. This deep permafrost may take centuries to respond fully to surface warming, providing a natural brake on the speed of feedback acceleration. The vegetation component of the feedback system also contains moderating elements. As Arctic ecosystems shift and adapt to changing conditions, some may become more efficient at capturing and storing carbon, potentially offsetting some of the releases from thawing permafrost. The balance between these competing processes, the release of ancient carbon and the capture of new carbon by expanding vegetation, remains one of the great uncertainties in understanding the Arctic's feedback symphony. Standing again on the Arctic tundra, as the light shifts from day toward evening and the great silence settles over the landscape, we can begin to hear the feedback symphony not as a source of anxiety or alarm, but as a profound conversation between the ancient and the modern, between the patient rhythms of geological time and the more rapid pulse of contemporary change. This symphony reminds us that the Earth climate system is not a simple, linear mechanism, but a complex, interconnected web of relationships where each component responds to and influences all the others. The methane rising from thawing permafrost carries with it the memory of ancient forests and grasslands, the history of past climates, and the patient work of countless microorganisms. Its release into the modern atmosphere represents not just a chemical process, but a conversation between different eras of Earth's history. The feedback loops that govern this process operate with a logic that transcends human timescales, responding to changes with the patience of ice ages and the persistence of tectonic drift. Yet they also respond to the more immediate influences of contemporary climate change, creating a bridge between the deep time of geology and the rapid time of modern atmospheric chemistry. In the end, the Arctic's Great Feedback Symphony teaches us about the interconnectedness of natural systems and the patient, persistent nature of planetary change. It reminds us that the atmosphere we breathe today carries the whispers of ancient swamps and the breath of long-dead forests, transformed by countless generations of microorganisms into the gases that help regulate our planet's temperature. As we listen to this ancient music, we might find in it not cause for despair, but reason for wonder at the intricate complexity of the world we inhabit. The Arctic's Feedback Symphony plays on, patient and persistent, a reminder that we are part of a conversation much larger and older than ourselves, and that understanding our role in this conversation is perhaps the most important listening we can do. In the vastness of the Arctic, where human presence feels like a whisper against the enormity of ice and sky, scientists have learned to become patient listeners, translating the subtle language that the Earth speaks through chemistry and physics. Their work resembles that of naturalists from centuries past, but instead of sketching birds or pressing flowers, they collect invisible gases, measure minute temperature changes, and decode the molecular stories written in ancient ice. The art of reading nature's signals in the Arctic requires a particular kind of attention, one that can perceive meaning in data points scattered across years and significance in changes measured in parts per million. Like astronomers studying distant stars through telescopes, Arctic researchers peer into the planet's atmospheric processes through instruments that can detect the faintest chemical whispers rising from the tundra. These scientific conversations with the Arctic landscape unfold across research stations scattered throughout the circumpolar north, each one a careful listener in the great atmospheric symphony we explored earlier. At places like Barrow in Alaska, Alert in Canada, and New York Alison in Svalbard, researchers maintain continuous vigils, their instruments sampling the air every few minutes, building long-term records that reveal the slow, patient changes occurring in Arctic atmospheric chemistry. The instruments themselves have evolved to match the Arctic's demanding conditions, designed to operate reliably through months of darkness and extreme cold, through summer's midnight sun and winter's savage storms. Automated gas chromatographs analyze air samples with the precision of molecular accountants, tracking the concentrations of methane, carbon dioxide, and dozens of other trace gases with an accuracy that would have seemed magical to earlier generations of scientists. But perhaps the most remarkable aspect of this scientific listening is how it reveals patterns that would otherwise remain invisible to human perception. The daily breathing of the biosphere, as photosynthesis and respiration create subtle rhythms in atmospheric carbon dioxide. The seasonal pulses of methane release as Arctic soils warm and cool. The gradual, year-over-year increases in greenhouse gas concentrations that mark our planet's changing atmospheric composition. At research stations like the one at Point Barrow, scientists have maintained continuous atmospheric measurements for more than 4 decades, creating one of the longest and most detailed records of Arctic atmospheric change. The data reveals patterns within patterns. The predictable seasonal cycles overlaid with longer-term trends, interrupted occasionally by the signatures of distant volcanic eruptions or the chemical fingerprints of major forest fires carried on Arctic winds. The process of collecting these atmospheric samples requires a kind of choreographed precision. Air intake lines stretch high above the research station buildings, positioned to avoid contamination from human activities. The samples flow through carefully calibrated systems that strip out water vapor, filter out particles, and deliver precise aliquots of air to the analytical instruments waiting in climate-controlled laboratories. Each measurement becomes a single note in a vast atmospheric composition, meaningful primarily in relation to all the measurements that came before and those that will follow. The concentration of methane in a single air sample, taken on a particular Arctic morning, gains significance only when compared to measurements from the same location taken years earlier or to simultaneous measurement from other locations around the globe. This network of atmospheric monitoring extends far beyond individual research stations, encompassing aircraft that carry instruments through different layers of the atmosphere, ships that sample marine air masses, and satellites that peer down from space to measure atmospheric composition over vast areas. Together, these diverse platforms create a three-dimensional picture of how greenhouse gases move through the atmosphere, where they originate, and how their concentrations change over time. The aircraft missions deserve particular attention for their ability to capture atmospheric processes in motion. Research planes equipped with sophisticated instruments fly regular patterns over Alaska and northern Canada, sampling air at different altitudes and locations to map the three-dimensional distribution of methane and other gases. These flights often reveal dramatic contrasts: higher methane concentrations at some altitudes and locations, lower at others, painting a picture of atmospheric mixing and transport that would be invisible from ground-based measurements alone. Satellite measurements add another dimension to this atmospheric detective work, providing the broad spatial perspective that complements the detailed temporal records from surface stations. Instruments aboard satellites can detect methane concentrations averaged over large areas, revealing regional patterns of emission and transport. While satellite measurements lack the precision and temporal resolution of surface stations, they excel at showing the big picture, how atmospheric composition varies across continents and ocean basins. The interpretation of these diverse measurements requires sophisticated computer models that simulate atmospheric transport and chemistry. Scientists feed their observational data into these models, which then help explain how gases released at the surface move through the atmosphere, how they mix with air masses from other regions, and how chemical reactions alter their concentrations over time. The models serve as translators, helping scientists understand the stories hidden in their atmospheric measurements. One of the most fascinating aspects of this scientific listening involves isotopic analysis, the study of different molecular variants of the same gas. Methane molecules, for instance, can contain different isotopes of carbon, and the ratio of these isotopes provides clues about the gas's origins. Methane from biological sources carries a different isotopic signature than methane from geological sources, allowing scientists to distinguish between emissions from thawing permafrost and those from natural gas leaks or other industrial sources. These isotopic measurements have revealed surprising details about Arctic methane sources. While much attention focuses on permafrost thaw, isotopic analysis shows that Arctic methane emissions come from a complex mixture of sources, some ancient, some modern, some biological, some geological. The relative contributions of these different sources vary from place to place and season to season, creating a complex tapestry of methane origin stories written in the language of molecular isotopes. Field research in the Arctic also involves more direct measurements of methane emissions from the landscape itself. Scientists use portable instruments to measure gas fluxes from soil surfaces, allowing them to identify specific locations where methane release is particularly active. These measurements often reveal surprising heterogeneity, areas of very high emission right next to areas where little or no methane escapes, reflecting the complex interplay of soil conditions, moisture, temperature, and microbial activity that controls gas production and release. Some of the most dramatic field measurements involve methane emissions from Arctic lakes and wetlands. Researchers paddling across thermokarst lakes, those formed by thawing permafrost, sometimes encounter areas where methane bubbles rise so vigorously from the lake bottom that they create visible disturbances at the surface. These bubble plumes, detected using underwater acoustics and direct gas sampling, provide windows into the active decomposition processes occurring in lake sediments. The seasonal timing of these emissions tells its own story about the relationship between climate and Arctic methane release. Peak emissions often occur not during the warmest months, But during the spring thaw, when warming temperatures first penetrate the frozen soil, and when water from melting snow creates the anaerobic conditions that favor methane production. This seasonal pattern, repeated year after year across thousands of measurement sites, helps scientists understand how Arctic methane emissions might respond to changes in the timing and intensity of seasonal warming. Long-term monitoring also reveals the influence of year-to-year climate variability on Arctic methane emissions. Particularly warm summers show up in the methane record as years of elevated emissions, while cooler summers correspond to lower releases. These correlations between temperature and methane release provide crucial insights into how Arctic emissions might respond to future climate change, offering a kind of natural experiment in the relationship between warming and permafrost carbon release. Perhaps most importantly, this extensive monitoring network has documented the gradual increase in Arctic methane emissions over the past several decades. While methane concentrations have risen globally, measurements from Arctic stations show that northern high-latitude sources have contributed disproportionately to this increase. The data suggests that the Arctic's contribution to global methane emissions has grown as warming temperatures have accelerated permafrost thaw and extended the growing season for methane-producing microorganisms. The precision required for these measurements pushes the boundaries of analytical chemistry. Detecting changes in atmospheric methane concentrations requires instruments capable of measuring differences of just a few parts per billion, equivalent to detecting a few drops of water in an Olympic-sized swimming pool. Maintaining this level of precision over decades of continuous operation through Arctic storms and equipment failures represents one of the quiet triumphs of environmental science. Quality control for these measurements involves constant cross-calibration between instruments and regular analysis of reference gases with precisely known compositions. Scientists regularly ship samples between laboratories around the world to ensure that measurements made in Alaska are directly comparable to those made in Greenland or Siberia. This international coordination allows researchers to build global pictures of atmospheric change from thousands of individual measurements collected under vastly different conditions. The human dimension of this scientific listening should not be overlooked. Behind every data point lies the work of researchers who have chosen to spend significant portions of their careers in some of Earth's most remote and challenging environments. Arctic field work demands a particular combination of technical expertise and physical resilience, as scientists maintain sophisticated instruments in conditions that can disable vehicles and shut down communications. Research stations in the Arctic often take on the character of small communities, where scientists from different disciplines and nationalities work together for months at a time, united by their shared fascination with Arctic processes. The collaborative nature of this work reflects the interdisciplinary character of atmospheric science itself. Understanding Arctic methane emissions requires expertise in microbiology, soil science, atmospheric chemistry, climatology, and numerous other fields. The data collected through this extensive monitoring network doesn't remain locked away in scientific archives, but feeds into the climate models that inform our understanding of future climate change. Atmospheric measurements from the Arctic provide crucial benchmarks for testing and improving these models, helping scientists understand whether their predictions about future methane emissions are consistent with observed trends in Arctic atmosphere chemistry. As this vast scientific conversation with the Arctic atmosphere continues, new technologies constantly expand the range of signals that researchers can detect and interpret. Advances in laser spectroscopy allow for more precise measurements of trace gases, while improvements in satellite instruments provide better spatial and temporal resolution of atmospheric composition. Each technological advance opens new windows into the atmospheric processes that connect Arctic landscapes to global climate systems. The story told by decades of Arctic atmospheric monitoring is one of gradual change, punctuated by seasonal rhythms and year-to-year variations. It's a story written in the language of parts per million and isotope ratios, but it speaks to the fundamental question of how our planet's climate system responds to changing conditions. In the patient accumulation of these measurements, scientists are documenting one of the most significant environmental changes of our time: the awakening of the Arctic's vast carbon reserves and their growing conversation with the global atmosphere. In the deep Arctic night, when the aurora borealis paints ethereal curtains across the star-filled sky, and the only sounds are the gentle creaking of shifting ice and the distant call of a snowy owl, there exists a profound peace that seems to transcend the complexities of atmospheric chemistry and climate change. Here, under the vast dome of the polar sky, the processes we have explored throughout our journey continue their patient work, largely invisible to human perception but fundamental to the planet's atmospheric rhythms. The Arctic night offers a unique perspective on the natural cycles that govern our world. In the absence of industrial noise and urban light pollution, surrounded by landscapes that remain largely unchanged from their ancient forms, one can sense the deeper rhythms that have shaped Earth's climate for millions of years. The methane molecules rising silently from thawing permafrost join an atmosphere that has witnessed the advance and retreat of countless ice ages, the slow drift of continents, and the patient evolution of life itself. Standing on the frozen tundra under these stars, breathing air so pure it seems to crystallize in the lungs, we might reflect on the extraordinary journey that has brought us to this moment of understanding. The ancient organic matter, now decomposing and warming soils, represents the accumulated life of countless growing seasons from eras when the Arctic climate was fundamentally different from today. Vast forests once grew where now only low shrubs and grasses survive, and the remains of those ancient ecosystems now fuel the microbial processes that contribute methane to our modern atmosphere. The stars themselves, burning with nuclear fire across the cosmic distances, provide a humbling perspective on the timescales involved in planetary change. The light reaching our eyes left those distant suns years or decades or centuries ago, traveling across space while the Arctic permafrost slowly accumulated its stores of organic carbon. The carbon now being released as methane was captured from the atmosphere by photosynthesis powered by starlight that began its journey when the climate patterns of Earth were configured very differently than they are today. This cosmic perspective reminds us that climate change, while significant on human timescales, represents just one movement in a vast symphony of planetary evolution that has been playing since Earth first developed its atmosphere billions of years ago. The current warming and the Arctic's response to it will eventually take their place in the geological record alongside the signatures of past climate changes that have shaped the planet's history. Yet this long-term perspective need not diminish the significance of contemporary changes or our responsibility to understand and respond to them. Rather, it can provide a framework for understanding current climate change as part of Earth's ongoing evolution, influenced now by human activities in ways that are unprecedented in the planet's history. The Arctic's response to warming temperatures represents a conversation between ancient processes and modern influences, between the patient rhythms of geological time and the more rapid pace of contemporary environmental change. The silence of the Arctic night also offers space for contemplating the remarkable nature of scientific understanding itself. The fact that we can stand on the tundra and comprehend the invisible processes occurring beneath our feet, the patient work of microorganisms, the gradual thaw of permafrost, the molecular journey of methane from soil to atmosphere, represents one of humanity's greatest achievements. Through careful observation, patient measurement, and collaborative analysis, scientists have learned to read the subtle signals that the Earth sends through its atmospheric chemistry. This understanding has emerged through the dedicated work of researchers who have chosen to spend significant portions of their lives in remote Arctic locations, maintaining instruments through long polar nights and harsh weather conditions, collecting data that individually seems insignificant but collectively reveals the patterns that govern planetary atmospheric chemistry. Their work represents a form of devotion to understanding that mirrors the patient, persistent nature of the natural processes they study. The methane molecules rising from the soil beneath our feet will join atmospheric currents that will carry them around the globe, mixing with air masses from tropical forests and industrial cities, participating in chemical reactions driven by sunlight, and interacting with countless other molecules in the vast atmospheric system that envelopes our planet. Each molecule carries with it a history of biological processes, a record of the conditions that led to its formation, and a future role in the planet's energy balance. The interconnectedness revealed by this molecular journey reflects a fundamental truth about Earth's climate system: that changes in one location can have consequences that ripple through the entire global system. The methane released from Arctic permafrost contributes to atmospheric warming that affects weather patterns, precipitation, and temperatures in regions far from the Arctic. The atmosphere serves as a medium of communication between Earth's different climate zones, carrying chemical messages that link the fate of tropical rainforests to Arctic ice and Arctic permafrost to global sea levels. Under the Arctic stars, we might also reflect on the relationship between knowledge and wonder. Scientific understanding of atmosphere processes, rather than diminishing the sense of awe inspired by the natural world, can deepen our appreciation for the elegant complexity of planetary systems. The fact that microorganisms in Arctic soil can influence global atmospheric chemistry, that ancient swamps can speak to us across millennia through their molecular legacy, that human activities can participate in planetary-scale atmospheric processes. These realizations enhance rather than reduce the sense of wonder that the natural world inspires. The Arctic landscape itself embodies this relationship between ancient and modern, between the patient rhythms of geological time and the more rapid pace of contemporary change. The permafrost beneath our feet may be thousands of years old, but it is responding to atmospheric changes that have occurred primarily within the last few decades. The balance between permanence and change, between stability and transformation, plays out in every aspect of the Arctic environment, from the slow advance and retreat of glaciers to the rapid seasonal changes in vegetation and wildlife. The northern lights dancing overhead provide their own metaphor for the processes we have explored. The aurora results from interactions between solar particles and Earth's magnetic field and atmosphere, invisible forces made visible through their effects on atmospheric gases. Similarly, the methane releases from thawing permafrost represent invisible processes made detectable through their chemical signatures in the atmosphere. Both phenomena remind us that the seemingly empty space around us is actually filled with dynamic interactions and ongoing transformations. The seasonal rhythms that govern Arctic life also offer insights into the nature of natural change. The dramatic transitions between summer and winter, between months of continuous daylight and continuous darkness, between frozen and thawed conditions, demonstrate that dramatic change is itself a fundamental characteristic of Arctic environments. The current climate changes, while unprecedented in their specific character, occur within a landscape that has always been defined by transformation and transition. Perhaps most importantly, the Arctic night sky reminds us of our place within the larger cosmic context. The atoms in our bodies were forged in stellar nucleosynthesis, making us literally made of star stuff. The oxygen we breathe was produced by billions of years of photosynthesis. The carbon in our cells has participated in countless cycles through the atmosphere, biosphere, and geological reservoirs. We are not separate from the natural processes we study, but participants in them, connected through chemistry and physics to the atmospheric systems that govern planetary climate. This connection carries with it both privilege and responsibility. Our species has developed the intellectual capacity to understand atmospheric chemistry, to predict the consequences of different emission scenarios, and to make choices about how our activities influence planetary systems. This understanding comes with an obligation to use our knowledge wisely, to consider the long-term consequences of our actions, and to recognize our role as participants in rather than masters of Earth's climate system. The Arctic teaches patience. The processes that govern permafrost thaw, methane release, and atmospheric change operate on timescales that dwarf human planning horizons. Yet the Arctic also demonstrates that persistence and dedication can yield profound insights. The decades of atmospheric monitoring that have revealed Arctic climate change patterns required sustained commitment and careful attention to detail. The scientific understanding that allows us to comprehend atmospheric processes has emerged through the patient accumulation of observations and measurements across many years and many research locations. As we prepare to leave this frozen landscape and return to more temperate environments, we carry with us an enhanced appreciation for the interconnectedness of planetary systems and the remarkable nature of scientific understanding. The Arctic has served as our laboratory for exploring atmospheric chemistry, but the lessons learned here apply to climate processes occurring around the globe. The patient work of microorganisms, the gradual accumulation and release of atmospheric gases, the complex feedbacks between biological and physical systems. These processes occur in different forms in all of Earth's climate zones. The methane molecules that will continue rising from Arctic soils long after we have departed will join the global atmospheric circulation, contributing to the ongoing conversation between Earth's surface and its atmosphere. Some will be broken down by chemical reactions in the stratosphere. Others will be absorbed by soil bacteria in distant locations, and still others will continue to trap heat in the atmosphere, contributing to the ongoing warming that drives continued permafrost thaw. The Arctic's patient breathing continues, largely invisible but fundamental to planetary atmospheric chemistry. The ancient organic matter preserved in permafrost will continue its gradual return to the atmosphere, mediated by microorganisms that have waited millennia for the right conditions to resume their work. The feedback loops between warming temperatures and methane release will continue to operate, responding to changes in global climate with the persistence of geological processes, but the relevance of contemporary environmental challenges. Under these Arctic stars, in this landscape of ice and silence, we have gained insights into some of the most fundamental processes that govern our planet's climate. The knowledge we have acquired carries with it a responsibility to share these insights, to use our understanding to make informed decisions about humanity's relationship with planetary systems, and to maintain the kind of patient, careful attention to natural processes that has made this understanding possible. The Arctic night embraces us with its silence and its stars, reminding us that we are temporary visitors in landscapes that have existed for millennia and will continue to exist long after we are gone. Yet our brief presence here has allowed us to glimpse the elegant complexity of atmospheric chemistry, to understand our connection to planetary systems, and to appreciate the remarkable nature of the scientific knowledge that makes such understanding possible. In the end, this may be the Arctic's greatest gift, not just insights into climate processes, but a deeper appreciation for the wonder and complexity of the world we inhabit.